Hi, my name is Dr. Jordan Whitlock. I'm a PhD in genetics, and I'm a member of the SETBP1 Society's Scientific and Medical Advisory Board. Today, I'm here to bring your second installment of the SETBP1 Science Bites, where we will be discussing the type of variants that are found in SETBP1 haploinsufficiency disorder. If you did not have a chance to, take a look back at our first webinar, where we cover what is SETBP1-HD in order to gain a basic understanding of genetics and how changes in the DNA are impacting our community. A variant is sometimes called a mutation and changes the DNA sequence of a gene in a way that makes it different from most people's. The SETBP1-HD population represents many different types of variant mechanisms. These can include substitutions, which are further divided into missense and nonsense, or frame shifts, which include insertions and/or deletions. These can range in size from a single amino acid or letter to covering multiple genes. One can check out how these variants are represented across the SETBP1 Society using the website's dashboard that's linked in the comments section of this video. There are many types of variants and mechanisms that cause them. One thing to note is that these changes are only occurring in one copy or allele of the SETBP1 gene. To understand how these different changes in the DNA are occurring and how they may be altering the SETBP1 gene, I'm going to walk through a quick example of the four most commonly seen types that we see today in our SETBP1-HD community using an example sentence on the graphic that you'll see now on the screen. So let's let the SETBP1 DNA, which is represented as a genetic code with the four letters A, T, C, and G, be represented by the sentence, "The fat cat ran off." When different variants are introduced, such as a substitution, in this case a missense in the first scenario, we see that a different letter or amino acid is being used that changes the meaning of the sentence to now be "the fat rat ran off." It still makes sort of sense and produces a somewhat functional SETBP1 protein, but it doesn't work or mean the same thing as the original or normal sentence. Next, we see different types of frame shifts, such as insertions or deletions, where there's either an addition of a letter to our sentence or a missing letter from our sentence that shifts the sentence around so that it doesn't make sense and is unable to produce a functional protein. In this case, deletions, for example, can range from a single amino acid or letter to covering multiple genes or large portions of the SETBP1 gene. The final variant that we would like to highlight is a nonsense mutation. These are also called, or you may have heard them referred to as premature stops. In the example with the premature stop, we see that the sentence stops abruptly, only producing "the fat" instead of our entire phrase that we started with at the beginning. In the same way, they likely see that the protein is truncated or not there at all whenever a nonsense mutation is introduced. The American College of Medical Genetics and Genomics, or ACMG, has developed a system to classify these different genetic variants into 5 categories based on their potential impact on health. These categories are shown on the screen, and they are as follows: pathogenic, likely pathogenic, variants of uncertain significance, which you may have heard of as a VUS or VUS, likely benign, and benign. However, these last two you may not have heard of because they typically aren't shown or included on a genetic report because they tend not to have a lot of clinical impact. These classifications help geneticists and healthcare professionals interpret the genetic results from individuals and provide guidance on management and treatment of different genetic conditions, such as SETBP1-HD. They're based on various lines of evidence that you'll see on the screen now, including population data, functional studies, and clinical observation. It's important because this information allows us to understand how the variant could be potentially clinically relevant. More information on this particular topic is provided by ClinGen in one of their resources titled An Intro to Genome Analysis. In SETBP1-HD, as of early 2024 to date, 59% of individuals have a pathogenic, 24% have a likely pathogenic, and 16.3% of SETBP1-HD individuals have a variant of uncertain significance. If you're interested in looking at the breakdown of variants and their clinical significance, check out the dashboard on the SETBP1 Society webpage. While in this video we focus on the 4 main types of variants that are seen in SETBP1-HD, we do not cover other types of variants today, including duplications, repeat expansions, intronic variants, and others. Thank you so much for listening, and let us know what you would like featured in the next SETBP1 Society Science Bite. Until next time.